Toxic bacteria linked to ulcerative colitis. A new understanding of inflammatory bowel disease. Ulcerative colitis, a debilitating inflammatory bowel disease, IBD, may be triggered by toxins produced by common bacteria found in contaminated water. New research from Nanjing University in China suggests that exposure to these bacteria leads to the destruction of crucial immune cells in the colon, significantly increasing the risk of developing the condition. This discovery could reshape how we approach both treatment and prevention of ulcerative colitis, which affects millions worldwide. The role of macrophages in gut immunity. Ulcerative colitis is characterized by chronic inflammation of the colon and rectum, often cycling between symptom-free periods and severe flare-ups. The disease's exact causes have remained elusive, despite being partially linked to autoimmune responses, genetics, and environmental factors. Researchers now believe macrophages, immune cells responsible for clearing debris and regulating inflammation, are key to understanding the disease's progression. Macrophages act as gatekeepers, sounding alarms when inflammation is needed, but also signaling a return to calm. Studies reveal that patients with ulcerative colitis have significantly fewer macrophages in their colon tissue. Experiments on mice confirmed that eliminating these cells made them more susceptible to developing colitis, suggesting that macrophage depletion directly contributes to gut inflammation. Aerolicin, the toxin at the heart of the problem. The critical link emerged when researchers identified aerolicin, a potent toxin produced by Aeromonas bacteria. This toxin selectively kills macrophages without harming other gut cells. Aeromonas strains capable of producing aerolicin, dubbed MTB, macrophage toxic bacteria, are frequently found in freshwater and brackish environments. When mice were infected with MTB, their susceptibility to colitis increased dramatically. However, disabling the gene responsible for aerolicin production or neutralizing the toxin with antibodies prevented this effect. Strikingly, the toxin was detected in the stool samples of 72% of ulcerative colitis patients, compared to only 12% of healthy controls. A complex relationship, not a simple cause. While the findings are compelling, researchers emphasize that MTB isn't the sole cause of ulcerative colitis. The disease appears to be multifaceted, influenced by both genetic predisposition and other environmental factors. Some individuals may carry MTB without developing colitis, and the disease can occur independently of this bacterial exposure. Persistent MTB infection can induce a hypersensitive state in the colon, but this does not mean that every infected individual will develop colitis. The occurrence of colitis in this context is undoubtedly influenced by environmental and genetic factors, explains Zuna Zhang, lead researcher on the study. Future treatments and rising incidence. The research opens several avenues for new treatments, including neutralizing aerolicin with drugs, developing vaccines targeting the toxin or the bacteria, or using phage therapy to eliminate toxin-producing strains. Martin Kriegel of University Hospital Münster notes that the link between MTB and colitis may be particularly strong in regions like China, where MTB infections are common. Given the rising global incidence of IBD, understanding the role of environmental factors like MTB could be crucial for effective prevention strategies. The findings suggest that ulcerative colitis may be more closely tied to environmental exposures than previously thought. Further epidemiological studies are needed to confirm the link between MTB and the disease, but the current evidence points to a significant and potentially preventable factor in the development of this chronic inflammatory condition.